talk before the panel discussion. Charlie, yes, sorry. So Charlie will talk about the analysis and visualization of large-scale drug combination experiment with Chinese.
Okay, thanks so much to everyone for having me here today, and thanks for the conference organizers. So yes, this is me. I'm Charlie Wright. I'm a chemical biologist by training, and I work now in the Department of Computational Biology at St. Jude, doing some methods development in the Department of Computational Biology. So I'm going to talk to you about analysis and visualization of drug combination data in Chinese. Why drug combinations? Well, this is pretty intuitive, but I'll tell you anyway. Most diseases require drug combination therapy. Let's see if I can get this to advance. Okay, and combining drugs can tackle a lot of diseases that have otherwise poor responses. So for example, like drug resistance and cancer are things that can be tackled by drug combinations. The goal of combining drugs is to look for synergy. So this is a term you may have heard before. So what is it? Synergy is really just illustrated by this cartoon here. Suppose you have like a tumor sample and you have drug A reducing the tumor volume by say 25%, B reduces by 25%. If you add them together, you would expect a reduction of 50%. Synergy is basically when you have a greater than expected effect. So in this case, 75% reduction. And how are drugs combined? This is really important for the rest of the talk. So you might just think to yourself, you know, here's two drugs, is it good or not? But in practice, what you need to do is analyze these drug combinations with multiple concentrations. So for example, you might have several concentrations of each of these drugs. And this is really important because, for example, you take like an aspirin pill, you know, this is not just some random concentration. Each pill is a very very well known concentration amount of that drug. And when you combine them, you have to put them into like a matrix and combine them in this fashion here to see if they're truly synergistic or not. But currently in the field, what happens is that you have studies doing these combination screens in very small matrices. So something like 3 concentrations of one drug, 3 by the other, or non-square matrices, like 2 by 7, or sparse matrices. But what really is needed is something like this. We call this a dense matrix. So 10 concentrations of each drug to produce a really wealth of data to really understand what concentration is truly providing the most synergy. Okay, so we have developed a pipeline, kind of a framework. The first part I won't talk about is the experimental part, where we're able to produce these 10 by 10 matrices. But after that, how are they analyzed? So to analyze like drug combination data properly, there's many steps, you know, and some of this can be quite tricky. You have to do a lot of reformatting and perform normalizations to your controls, actually run the synergy calculations, refine with statistics if you have replicates. Most importantly, you have to visualize everything. And currently, there's not really a lot of usable tools that can do this. There are some here and there, but and there's a few private ones, but what's really needed are open source tools to really analyze synergy and visualize the results on a scalable method. And drug combination screens, I'll mention, are like in super high demand. Every institution is interested in drug combinations, but the tools to analyze and visualize them are not readily available. So what I've been working on is developing this framework called ComboCat, which is able to automate this kind of complex analysis and visualization with drug combination data. And the visualization part comes from Shiny. So we've had some really great speakers talk about this during this conference so far. Really, the goal of ComboCat is really to automate this by taking in a plate of raw data. So this is a standard 384 well plate used in all kind of biological experimental applications. So if you just have this completely raw data in a specific format that goes in hand with the experimental side and a simple metadata file just describing very simple terms, what we want to do is take all of that and perform a lot of really complex functions on that to get the synergy, and then visualize that all in Shiny. So I'll just mention some of the steps. This is kind of a broad overview, and if you see some of these like gray squares, these are the names of the functions that I've written for this. But basically what we want to do is map this data to tell it, I'm saying like drug 1 here is the upper triangle of one of these wells, drug 2 is the lower triangle, and it goes by concentration, low to high, blue to red. And we want to tell all of this in the back end, this is exactly this combination of which drug and which replicate, and then construct the replicate matrices from this. We call this the mapping step. After that, we normalize so that you have these 3 10 by 10 drug combination matrices. And now this is a function of percent cell death, something more intuitive, right? And very easy to understand. As you increase the concentration of each drug, you increase the percent cell death. We do things like fit dose response models and plot them, as well as extract the IC50 values, just take the averages of cell death. And then very importantly, one critical step in synergy analysis that you need to do is actually calculate the synergy from this as an input. So we do that and calculate the synergy for every single one of these 100 squares. And then we can plot something like the maximum synergy here. So this is like, if you look at this square here, this is the most synergistic out of this combination. So if you look at the respective drug-only values and then plot what they look like together, it makes it very easy to see what the true synergy is. So that was like a very broad overview, but perhaps you have many, many combinations and many samples as you really need to answer some very specific synergy questions. What we want to do is take all of this, apply it to all of the samples a user might input, and then analyze everything in a project level. So this is kind of a way to analyze everything together. I'm going to show you a very quick demo of how we do this. If you can still see my browser, hopefully. Okay, so here's just an example of this here, the ComboCat tool, where I've just clicked this button to load in some preloaded data, and you can see the metadata pops up. It's 9 different plates of combinations. If you click the analysis tab here, what you do is we have a drop-down list that can render all of this information for any sample that you choose. So some of these are very well-known synergistic combinations because this is example data we generated, where we have everything like from dose response matrices that are interactive, so you can see the replicate values here, percent cell death matrix, like I've mentioned, calculating the synergy, and then many other plots like I showed before, where something like this is especially striking, where you look at the concentration of drug 1, concentration of drug 2, and then combine them. So we can print out a table, and what I really want to drive the message is that all of this is done and rendered in Shiny. So it's a really fantastic way to explore this synergistic data. We can also do this composite analysis, which has analyzed all of our data collectively and look at the distribution, for example, of cell death or synergy for all of our samples, as well as rank every single sample. So we know out of all the thousands of data points we have, which compound was the most synergistic. So that's just kind of like a high level overview. So basically that's what I wanted to show. So the message I'll leave you with is that, ComboCat, we're finishing it up right now. We have a lot more features coming through, but it's almost finished. It's going to be completely free, public, open source. And making usable tools like this is really critical to overcome the barrier to some important problems. So with that, I'll leave it at that and just say thanks to a few people. The Gielaher Lab, which is where I work in at St. Jude. Thank you very much to the POSIT community that I ask questions to all the time, to the organizers here, and to really everyone. But I did want to point out just 3 really titans of education in R and Shiny that I follow very closely, and I recommend you do too. Julia Silgi, Dean Atali, and of course Eric Nance, who all of whom are very, very important in our community for educating. Okay, so I think that's it. I'm just going to click stop sharing, and thank you all for your time.